What is up, y'all? Welcome to PowerPoint Lab Animations. Today we are talking about a couple of blood bank tests, the direct antiglobulin test, or the DAT, and the indirect antiglobulin test, or the IAT. We're going to look at what's happening inside those test tubes when you're performing a DAT or IAT, but first we'll talk about why we perform these tests and what these results mean for our patients. As always, what you're going to see today was created and recorded in PowerPoint by a real person. So today we are talking about the DAT and IAT. Both of these tests use a reagent called anti-human globulin, or AHG, to detect IgG-coated red cells. The DAT detects red cells that were coated with antibody in vivo, and the IAT detects red cells that were coated with antibody in vitro. AHG is a solution of antibodies directed against human antigens, which is why it's called anti-human globulin. Different types of AHG are used at different stages and with various modifications of the DAT and IAT. So in the DAT, we are looking for cells that were coated with IgG in vivo. If you're an MLS or MLT student, you should be able to come up with a few scenarios where this can happen, but the big three are HDFN, WEHA, and DHTRs. In HDFN, IgG from the mother crosses the placenta and binds to fetal red cells, which leads to those cells being hemolyzed. In many hospitals, a sample of cord blood from every birth is sent to the lab for a DAT to screen for HDFN. In WEHA, or warm autoimmune hemolytic anemia, autoreactive IgG can cause severe extravascular hemolysis and anemia. In DHTRs, or delayed hemolytic transfusion reactions, patient antibody is directed against donor red cells, which are eventually hemolyzed in the spleen. Each of these conditions can potentially be detected with a positive DAT. The IAT is used to detect red cells that were coated with IgG in vitro. And we use this test in a few different ways, and this is where some students get confused. If you work in a blood bank, you will get orders for a DAT in the lab. If it's on cord blood, you're screening for HDFN. If it's part of a transfusion reaction workup, then you are trying to rule out a hemolytic transfusion reaction. You will not get orders for an IAT. Instead, you'll get orders for an antibody screen or a cross match, and you will perform an IAT to fulfill that order. Really, both the DAT and IAT are methods that are applied to various samples and in various scenarios, and the results mean various things in those various settings. But for whatever reason, convention demands that a DAT is always called a DAT, and an IAT is always called something different depending on how it's being used. The IAT is used almost exclusively in the pre-transfusion testing, and in this setting it has three applications worth mentioning here. The IAT is used to screen for clinically significant antibodies in recipient plasma, aka an antibody screen. It's also used to phenotype donor or recipient red cells for clinically significant antigens, aka antigen typing, or to ensure the viability of donor red cells in recipient plasma, aka cross-matching. A patient with a positive antibody screen that needs blood may have more than a dozen IATs performed in the name of getting them compatible units. Last thing before we get to the animations, I do want to talk a little bit about the reagent that unites these two methods, which is anti-human globulin, or AHG. There are all different types of AHG depending on what you're using it for, but in a standard DAT or IAT, the AHG used will be a polyspecific blend of antibodies, meaning the antibody solution can detect multiple antigens or it has antibodies of multiple specificities. A polyspecific AHG will detect C3G and the Fc portion of IgG. The reason this reagent is necessary is because IgG in complement cannot agglutinate red cells on their own the way IgM antibodies do. You can see why that is in the image up here that I borrowed from Pearson. This boils down to the fact that the pentameric IgM molecule is able to bridge zeta potential and the monomeric IgG is not. But just because they are not complete agglutinins in vitro doesn't mean they won't lead to phagocytosis by splenic macrophages in vivo. We kind of take it for granted now, but before the discovery of AHG, there was no way to detect red cells coated with IgG in the lab. This reagent allowed the blood banker to visualize a whole new category of potentially hemolytic transfusion reactions before the blood is ever given. Now, on to the animations. We're going to start by looking at what happens when we add some IgM to a red cell suspension where the antigen is present. And this is what it looks like before we spin it down. Just some red cells and pentameric IgM floating there. When we centrifuge this, the IgM and red cells are forced together, and if the correct antigen is present, one molecule of IgM can bind several red cells. Those red cells can then bind more IgM, and that IgM can bind more red cells until you get a big clump of red cells and IgM, and this is what we call agglutination. This is what's happening in your tube when you're doing AVO typing, and it's why we can so easily visualize those reactions. Now let's look at what happens when we add IgG to a red cell suspension with the antigen present. When we spin it down, we end up with IgG-coated red cells, but the monomeric IgG can't bind another red cell, and so visually, you end up with nothing, or no agglutination. And this is a problem because if you actually have IgG-coated red cells in your tube, those cells are eventually going to be hemolyzed by the patient. The solution to this is to add AHG. The AHG is the green antibody floating here, and if you've used AHG before, that should make sense. The AHG is able to bind multiple Fc portions of IgG, so we end up with detectable agglutination. All right, we will wrap this video up by talking about how to interpret the results of a DAT or IAT. There are no reference ranges for these tests because they are qualitative and we would almost always prefer them to be negative. The interpretation really depends on the context. Positive cord blood DAT is an indication of HDFN, but a negative result does not necessarily rule it out. If you're evaluating a transfusion reaction, a positive DAT on the post-transfusion sample indicates the reaction could be due to a clinically significant antibody. For a patient with anemia and spherocytosis, a positive DAT can identify the cause of the patient's anemia. A positive IAT anywhere along the line in pre-transfusion testing indicates an incompatible donor-recipient combination. The antibody screen is the first step where recipient plasma is tested against a panel of O-negative reagent red cells. Agglutination in the antibody screen means that the recipient has a clinically significant antibody that could hemolyze donor red cells positive for the corresponding antigen. That antibody has to be identified by running a series of IATs testing recipient plasma against more reagent red cells. When the antibody is successfully identified, the blood banker then has to antigen type donor units to find cells that lack the corresponding antigen. That means running more IATs on donor units using reagent antibodies until you find enough negative units to fulfill the order. And then once we found some antigen negative donor units, we have to cross-match them, which is another IAT, where we test the antigen negative donor units against the recipient plasma just to make sure there's no funny business that we missed in the previous testing. Agglutination at the cross-match stage means the units are incompatible with the donor. No agglutination means no potentially hemolyzing antibodies were detected. And that's going to be it for today. Hope you enjoyed the video. Let us know below if there are any other lab tests or lab-related concepts that you would like to see animated in PowerPoint. Thanks.